Topic: Genetics, pedigree diagrams, and genetic engineering. Aims of lesson. At the end of this lesson, you should be able to know the following: genetic lineages/pedigree diagrams, mutations, genetic engineering, paternity testing, and genetic links.
Introduction. Revise the topic meiosis as covered in the week 4 and 5 lessons. Revise the concepts of inheritance, genetic crosses, sex determination and sex-linked inheritance. Covered in the week 6 lesson. Concepts and skills. Study the following information. Pedigree diagrams. A genetic lineage/pedigree traces the inheritance of characteristics over many generations. Example. The pedigree diagram below shows inheritance of eye color in humans over 3 generations of a family. Brown eye color, B, is dominant over blue eye color, B. Squares represent males and circles represent females. The horizontal line between a square, Joshua, and a circle, Ronel, shows that they have mated. The vertical line flowing from the horizontal line represents the offspring, Sarah and Peter, of the 2 parents, Joshua and Ronel. Remember the following steps when interpreting pedigree diagrams. Step 1. Study any key and opening statement/s and look for dominant and recessive characteristics and phenotypes. Brown eye color, B, is dominant over blue eye color, B, as stated in the problem. Step 2. Write in the phenotypes of all the individuals as given in the problem. Joshua, Jack and John are males with blue eyes. Veronica and Marlena are females with blue eyes. Peter and Frank are males with brown eyes. Ronel, Sarah and Gail are females with brown eyes. Step 3. Fill in the genotype of all the individuals with the recessive condition. It must have 2 recessive alleles, 2 lowercase letters, e.g. BB. Joshua, Veronica, Marlena, Jack and John will have the genotype BB. The recessive characteristic only shows up in the homozygous condition. Step 4. For every individual in the diagram that has the recessive condition, it means that each allele was obtained from each of the parents. Work backwards and fill in 1 recessive allele for each parent. Step 5. If the parents showed the dominant characteristic, fill in the second letter which represents the dominant allele, a capital letter, e.g. B. The genotype of Peter is BB, working backwards from the offspring Marlena or Jack or John who are homozygous recessive. This means that one of the recessive alleles of Marlena, Jack and John, i.e. B, must have come from parent Peter and the other one from parent Veronica. Step 6. Any other individual showing the dominant characteristic will most likely be homozygous dominant, BB, or heterozygous dominant, BB. Ronel could be homozygous dominant, BB, or heterozygous dominant, BB. Mutations. A mutation is caused by a permanent change to the DNA of a cell. Harmful mutations cause changes in DNA that can cause errors in protein sequencing, that can result in partially or completely non-functional proteins. Harmless mutations occur in the non-coding DNA and have no effect on the structure or functioning of the organism. Useful mutations can be advantageous to the organism and they are passed on from parent to offspring. These mutations also change the DNA responsible for the production of a specific protein. If the protein made increases the organism's chance of survival, it would be seen as a useful mutation. Gene mutation. A gene mutation is a change in the sequence of nitrogenous bases/nucleotides in a gene and can occur during DNA replication, transcription and crossing over. Examples of gene mutations are hemophilia and color blindness. Hemophilia, absence of blood clotting factors. Color blindness, due to absence of the proteins that comprise either the red or green cones/photoreceptors in the eye. Chromosomal mutation. Chromosomal mutations occur when meiosis does not occur normally. It may result in a change in the number or structure of chromosomes, e.g. during anaphase I if the chromosomes do not separate. Both chromosomes go to the same pole, and thus the chromosome number of the gametes changes. An example of a chromosome mutation is Down syndrome, due to an extra copy of chromosome 21 as a result of non-disjunction during meiosis. Genetic engineering. Genetic engineering is the manipulation or transfer of genes from one organism to another to make products that satisfy human needs. Genetically modified organisms, GMOs, are the result of genetic engineering. Example: genetic engineering in medicine. Synthetic insulin is used to treat diabetes and is produced by genetic engineering technology. See diagram and explanation below. Example: genetic engineering in medicine. The DNA with the gene coding for the production of insulin is removed from healthy human pancreatic cells. Enzymes are used to cut the DNA into segments to isolate the specific gene. A plasmid/circular DNA is removed from a bacterial cell. The plasmid is cut using enzymes. The human insulin gene is inserted into the plasmid to form recombinant DNA. The recombinant DNA is placed back into the bacterial cell. The bacteria are placed in a fermentation tank where they reproduce rapidly to produce many copies of the insulin gene. The insulin is extracted from the bacteria and purified. Advantages of genetic engineering. Produce crops that are resistant to adverse conditions/drought/disease/pests. Increase crop yield. Change the time for the ripening of fruit. Increase shelf life of plant products. Improve nutritional value of food. Improve the taste of food. Developing fruit/plants with desirable characteristics. Production of drugs or hormones, e.g. insulin, which have fewer side effects and is cheaper. Stem cell research. Stem cells are undifferentiated cells that have the ability to grow into any tissue in the body. They may be harvested from the placenta, embryos, bone marrow, and from blood in the umbilical cord. Use of stem cells. Bone marrow has been used for a long time to treat cancers. Stem cells are used to replace dead cells in the heart after a heart attack or growing skin tissue to treat burn victims or growing nerve cells to treat spinal cord injuries and Parkinson's disease. Cloning. Cloning is the process where genetically identical organisms are produced. Process of cloning in animals. The nucleus of an ovum is removed and replaced with the nucleus of a somatic donor cell/diploid donor cell. The zygote is stimulated for mitosis to occur. The embryo is then placed into the uterus of an adult female. Process of cloning in plants. Plants may be cloned by vegetative reproduction/asexual reproduction/tissue culture/grafting. A plant with the desired characteristics is selected. A vegetative part of the parent plant structure is removed and placed inside a growth medium and allowed to grow. Benefits of cloning. To produce organisms with desired traits. Conservation of threatened species. To create tissues/organs for transplant. Paternity testing. Blood grouping. The blood group of a child is determined by the alleles received from both parents. The blood group of the mother, the child and the possible father is determined. If the blood group of the mother and possible father cannot lead to the blood group of the child, the man is not the father. If the blood group of the mother and the possible father can lead to the blood group of the child, the man might be the father, but this is not conclusive because many men have the same blood group. DNA profiles. A child received DNA from both parents. The DNA profiles of the mother, child and the possible father are determined. A comparison of the DNA bands of the mother and the child is made. The remaining DNA bands are compared to the possible father's DNA bands. If all the remaining DNA bands in the child's profile match the possible father's DNA bands, then the possible father is the biological father. If all the remaining DNA bands in the child's profile does not match the possible father's DNA bands, then the possible father is not the biological father. Genetic links. Mutations in mitochondrial DNA is used in tracing female ancestry. Mitochondrial DNA, mtDNA, is found in mitochondria. Mitochondrial DNA mutates at a regular rate, so scientists are able to analyze these mutations to work out a timeline of genetic ancestry. Only the mother's mtDNA is passed on to her offspring. Analysis of mutations in mitochondrial DNA shows that the oldest female ancestors of humans are from Africa. Common errors made by learners in examinations. Unable to interpret pedigree diagrams. Do not know the definitions of genetic engineering, stem cells and cloning. Unable to explain how blood grouping and DNA profiling are used in paternity testing. Thanks for watching. Please do not forget to like, share and most important subscribe to the channel.